I want to introduce to you Dr. Stephen Gasior, who's going to speak about why we age and what to do about it. He's going to present current molecular and physiological models of aging and implications for treatment, regimens, and lifestyle. Based on these models, we can discuss how drugs, medicine, lifestyle activities, and even gene therapy would be useful based on each model. Stephen is a molecular biology scientist who was first introduced to Second Life while teaching biology at the University of New Orleans. His interest in virtual worlds was piqued when the distance learning team there offered a workshop in Second Life. It wasn't long before he's gotten himself an avatar and was looking at ways to use SL to teach biology. I taught 3 semesters of biology to non-biology majors in Second Life from the summer of 2010 to the spring of 2012, he says. I also developed several in-world activities and objects for virtual teaching of biology and chemistry. Wanting to do more than just lecture, he recreated and took students on Darwin's voyage aboard the Beagle, arranged a visit to Mendel's Abbey, and taught them the basics of genetics by having them breed virtual bunnies, using the affordances of Second Life just as they should be used. Currently, he's focusing his efforts on promoting science circle activities here in Second Life and outside, and working at Corteva Agrisciences. And I'd like to say welcome, Stephen.
Thank you, Wisdom, and thank you for the opportunity to present one of my scientific interests that are not in line specifically with my job, something from a bygone age of my molecular biology research. I'd like to welcome everyone to the talk. I think this is a great conference. There needs to be a lot more attention on aging well, and Whole Brain Health is a wonderful organization. We're helping people both physically and mentally try and age in the way that maintains happiness and health. This is also a great time for this type of talk and update because, one, of course, we are living in an aging world. The average age of people is increasing because of the advances of sanitation, medicine, overall wealth. The other aspect, though, is that really in the past, I'd say 15, maybe 20 years, there has been a tremendous amount of research at the very basic level that has given us a brand new understanding of the real basic physiology of what aging is from a very cellular and physiological level. And I think it's very important to update and talk about this because in a modern world where people are trying to make money, in many cases, they're trying to delude you into a solution that is not based on reality and it's not going to help you. So I think conveying the basics science of aging is really important for people understanding the context of what will and may not help in terms of healthy aging, especially when it comes to supplements or other things you might do. So I want to make a quick note that there is a box here on the stage, and that has both the conference program as well as a note card with the references for my talk. But those will both be made available on the conference website within the next week. And so let's begin. Overall, again, this is only going to be a 45-minute talk. It's going to be an overview to try and convey some basics. So in general, we'll talk about kind of how we define aging, especially when it comes to something in your basic science research labs. What are they measuring? How is that actually objectively figured out? And then I'll talk about what is the primary model for aging that scientists are working with and seem to have the most evidence and support for. And I'm going to talk about various drivers of aging. And I use that term specifically because I don't want to necessarily imply that any given one of them is the direct cause of aging. But we do know that they are associated with the aging process. And we have, within the models of aging, very specific reasons for why we think they are very important things that are driving the aging process. And based on how we are understanding those, I'll both show some examples in the scientific literature of interventions that alleviate those drivers of aging, and then kind of bring that back around at the end for the conversation we'll have about what this means for healthy aging in terms of what you might come across in the clinic, what the future of aging therapies might be, and what you may want to think about in terms of supplements. So I do want to make one other quick note too, which is especially if you're a researcher or a scientist or do some follow-up research on this, one thing I do tend to do in my talks is to bias my resources to those that are publicly available. That way, again, there are times where there are places I've worked or ways where I have specific access to things, but I want to make sure that the examples I give are something that are also available to you if you want to follow up on that information. And then the other bias sometimes is I try and find the best simple explanation and scientific evidence that also that where the original publication has a very nice, as many concepts as possible in one graphic so I can convey it to you. So there is a little bit of a bias that way. So if you're wondering why I may or may not talk about somebody's research, that might be why. So when we think about aging, a lot of it is I know it when I see it. And so we all can associate the prevalence of gray hair as something that comes to us as we age, skin getting a little more wrinkled, maybe a different gait or stooped shoulders. Again, these are very systemic, visible signs of aging that we understand are associated with people as they get older. But those tend to be a bit, in many ways, cosmetic. More importantly, in some cases, are hormone levels. What's actually happening in terms of our cells and our tissues and our physiology is that there are chemicals and chemistry that's actually changing. And so one example I have here is talking about testosterone, that as in particular men, as they get older, those testosterone levels decrease. And there are very important functional consequences to this, is that with the decreasing in testosterone, you might lose a feeling of energy, you might lose some degree of sex drive, also the ability to exercise and put on muscle mass. These are things that have very functional consequences to how we live our lives. And then to that next level of consequence, on the lower left-hand side, I'm showing the mortality curves from a recent pandemic that swept around the world. And if you look at this graph, what you'll notice is that there was a very big bias in the distribution of fatality, and again, also the severity of effects from it, but that this disease susceptibility is something that is consistent among a lot of different pathologies as we get older. And so in addition to disease susceptibility, just like pneumonia, COVID, you can also think about things like cancer or age-related dementia. And so here's an example of this, is that when it comes to neurological impairment, this is something that has a high prevalence of effect and something that is increasingly prevalent in modern day society. There may be things that are driving neurological impairments, pathologies due to lifestyle or the environment. And so I believe the current estimates in the Western world is that by the age of 65, 10% of people will develop Alzheimer's. By the age of 85, that's somewhere in the range of 30%. And there is a very, very big cost burden to things like Alzheimer's and Parkinson's diseases. There is certainly a decrease of enjoyment and mobility and activity within your own life. And then the medical bills and societal cost of that is very large and highly consequential. Another aspect too is as you get older, depending on what things have happened to you, there's this idea of frailty. Again, you can a lot of people when you think of some of them as being old, it's because you're seeing them with a slow gait, maybe a cough, they're stooped, they're very slow to react, maybe they're even shaking a little bit. In my own experience, they tend to be the ones driving the slowest in the left lane. And so that frailty is something that, again, has an incredible amount of impact on the enjoyment and activity that one has in their life. And what I want to show here in particular on the slide is the attempts to, from a whole body point of view, try and find ways to measure objectively aging. And so again, in terms of the specific aspect of what we call frailty index, is you have several survey questions, you observe people, you have put them on a treadmill, watch them walk, and you measure a lot of stuff to get an objective sense of what is their frailty index, which we know is then related to the aging process. And of course, once one can start developing these indices, these ways of measuring it, then you can also measure whether interventions that you're trying to help with are improving that. So that is a bit of the whole body perspective on aging and the physiological consequences. As a molecular biologist, I want to make sure I convey, and what the field is doing, is the cellular level of phenotypes that we associate with the aging process. And so again, I'm not per se as of yet talking about what is a cause of aging here, but we do know that when we look at the cells of people who are older, we see hallmarks of they have fewer stem cells, and stem cells are the important cells to replenish tissues within our body. Some of those cells are slowing down. They're becoming what's called senescent. They're not doing much, but they're still there. They might have other energy or regulation or chemical compound abnormalities. So things related to mitochondria, which makes energy for the cell, how well their DNA is packaged, whether their proteins are working or not. And then also the inability to recycle that. So the term autophagy is this ability to recycle the broken down components of the cell so that you can then clean up the clutter and make fresh ones. Another one example is inflammation. So inflammation is something like when you get a cold, of course, you feel that effect of inflammation of edema, water collecting, maybe fever, but there's also a cellular level at which inflammation occurs that has important consequences. So you might have heard of the terms interferon. And there may be lots of things that we still don't know that are important for understanding what a cellular age is like. Now, one of the things mentioned, of course, is telomeres, and this is something that has made a lot of popular press for a while. And so I do want to cover those a little bit specifically, because they also gave us insight back in the day to what it means for cells to age. And so the graphic I'm showing here is an example of, here you have a chromosome, and it's been duplicated. It's about to undergo division to make new cells. And those little orange caps at the end, those are called telomeres. That's because they're at the end, telo meaning distance. And they're important components of chromosomes because of the way that DNA replicates. It can't replicate the end of a chromosome. One part always shortens up. And that's why I use the term aglets. If you think about what would happen with shoelaces, your shoelaces can eventually unravel if it's missing that little aglet at the end, and it frays apart. And this is something that happens in all cells as they're dividing. And what's important to know is that stem cells or early embryonic cells, the ability to replenish telomeres is present. That's a compound known as telomerase. But what happens is most of our cells do not replenish the telomeres. Even our adult stem cells do not replenish telomeres. So there is, in essence, a limited amount of times cells can divide until those telomeres degrade away. And what I'm showing here on the bottom is a relatively recent review of this, a large-scale population assessment that's showing the length of telomeres. So the x-axis is the average length of telomeres within a sampled population. And the different colors represent different ages. And so shifted far to the left, the reddish orange are people who are centenarians. So they're very old, and that's also associated with shorter telomeres. And as the colors go more towards the blue and black range, those become younger. And so one thing that's to recognize here is that as we're growing, our telomeres get shorter, and those are at some level a marker of how many rounds of replication our cells have gone through as well. And so that's associated with what our age is. Now, speaking of the chemistry of the cell, there's another very important chemical marker known as methyl groups. And so you might have heard of the gas methane. It's a highly volatile compound, gas, which is a carbon and 4 hydrogens. And so that term methyl, that methylation group, is what you see on the top compound. Is you take a cytosine, and on the left, it's got that hexagon there, and on the right, you see in that red circle a methyl group added onto it. And so it's a very small change in the chemistry. This can still act as a DNA base pair, but it ends up being a marker of whether genes are turned on or off. And so when it comes to any given cell in our body, it's differentiated in a way where certain genes are supposed to be on, certain genes are supposed to be off, and that's what helps essentially create the identity. And in some cases, if we need to turn a gene on, then the ability to demethylate it is important. If we want to turn a gene back off, then you want to remethylate it. And so this is a very important marker for whether genes are on or off. And what's important that people discovered starting at the early part of the century, and I think it's most well characterized by Dr. Horvath, who's the last author in the citation, is they found that if you measure the methylation at very specific sites within humans, again, you have to pick these carefully, that these end up having very strong correlations to cellular aging, as well as, and this is the important part, predictive power. They actually give a measure of one's health span and can be predictive for lifespan. And so the important contrast here is that you might have heard of telomeres as an important thing for cellular aging, and they are. But in terms of their value of using them to understand clinically the cellular age of a person, methylation is actually the most reliable considered method for doing that. And in fact, there are methylation kits. If you ever wanted, you can now buy these. You can buy methylation kits to give you a sense of what your methylation clock age is. So within a generalized way of understanding how old cells are, that's a kind of generally useful way of looking at methylation. Once it comes time to the clinic or doing research models, there are other genes where you look at the expression of genes, whether they're on or off, that helps us get a sense of what's happening physiologically. So in terms of the larger sense, how do we model what is the aging process? I think this is the most currently accepted working model for understanding aging in the population. And the idea here is that when you think about evolution, you think about biology, it's very important that we get to our reproductive age, and then we reproduce and have, again, in cases where we, as a parent, are also a caregiver to our offspring, again, there's a lot of what's known as parental care, a high R value, that our bodies can survive and be around for that process. And then after that, it doesn't really matter too much. And so that's the disposable soma idea, is that, well, we have reproductive organs that are very important and the ability to raise our offspring. After that, there's not very much evolutionary action that's relevant and important for keeping us around longer. And so while we do go through developmental processes to get to reproductive age, it's not believed that there are reproductive pathways that then tell us to become older and more frail. So then why do we age? It's because we might be accumulating damage and clutter and problems in our cells because there's no reason not to, right? Eventually we're going to die from the accumulation of junk and clutter. And I think the key questions that people have been asking for the longest time, once we start understanding these processes and whether we're able to measure them, and now that we're able to measure them, is that reversible? And if it is reversible, is there a point of no return? In addition to understanding how we might actually do that reversing process. So that's what I'm going to go through here in the next series of slides. The bulk of my talk is going to be discussing, from a more specific point of view, what are the things that lead to this accumulation of damage, what leads to a loss of cell identity, what leads to the things that we now then associate from a full body point of view with the aging process. So one of the first models that was actually proposed I think way back in the 1940s, is the free radical theory of aging. And the idea here is that organisms age because they're doing chemistry. And when you're doing chemistry, you create reactive oxygen species, which then are partially reduced metabolites of molecular oxygen generated as products of metabolic reactions, and or as byproducts of various cellular processes such as respiration. Just the mere act of breathing, you're making reactive oxygen species. And those reactive chemicals, as you're looking at some of them here on the left, I have some examples of what are known as reactive oxygen species, that those then glom on and mess up the chemistry of other parts of the cell, either DNA or protein or the cell membrane and the lipids, and that ultimately leads to a breakdown of the cellular processes. And that is what drives aging. Now, one thing I want to point out here, the way I have my talk, again, in order to keep the slides a little bit more concise, sometimes it's very important to read the title of the citations at the bottom, because this is an example here where I got this from an article called "The Free Radical Theory of Aging is Dead." And that's because while this is a process that does occur in cells, there's not been a significant amount of body of research to suggest that this is a main driver of the actual process. So for example, taking antioxidant interventions has not been demonstrated to be a major way to intervene in the aging process. Now, there are things that are associated with reactive oxygen species, like mitochondrial dysfunction, that may be more correlated with as a driver of aging. But in terms of a fundamental process of aging, the free radical theory is not considered to be probably the major one anymore. Now, the overall idea that metabolism, however, is involved in aging, there's a lot of strong support for that. And again, if you look at people who have mutations or variations in growth factors or other hormones, you can actually see differences in lifespan. And so the example I want to give here, though, is one known as rapamycin, was originally discovered as an antifungal and then discovered in the clinic as a pretty useful immunosuppressant. And then interestingly, the names of these genes, TOR1 and TOR2, they actually got their name because they were discovered as the target of rapamycin. That's where the name comes from. And as these compounds were looked at and examined in the lab, as people try to understand what TOR1 and TOR do in mouse models, as well as even other models like Drosophila, the fruit fly, or nematodes like C. elegans, they discovered that they actually have the phenotypic consequence of extending lifespan. And so I'll give an example of that in the next slide. But merely giving model organisms rapamycin is able to basically slow down the aging process. And what I have here in the diagram is an example that these TOR1 and TOR2 proteins that are within these complexes, and what they do is they drive various cellular processes, or they inhibit various cellular processes. And so they can drive inflammation. They can drive lipid synthesis. They can inhibit or drive protein synthesis. And so what's important to recognize is that they exist as sensors of environmental metabolism, and then they drive the cell to do things that are responsive to that. And in fact, some of these things that drive these cellular processes are associated with aging. So again, it's a way of connecting metabolism to what the cells are doing. And so what this next slide is showing, again, the top slide, this is a very common way using model organisms to try and understand if you were intervening or changing the lifespan of an organism. And so these are, the x-axis here is the age of the mice in days. So mice on their own, lab mice tend to live on their own to about 3 years old, maybe only 2 years old, depending on what strain you have. And so the normal, if you look at a population of mice, they tend to follow this blue curve. Is that as they get older, more in the population start to die, and then that curve goes down. And what these graphs are demonstrating is that if you start feeding the mice rapamycin at an early age, within I believe 2 months of being born, then that red graph, that red curve is showing you that more of them are taking longer to die. And so that's an example of a lifespan extension curve. And so these effects tend to be stronger in females. Why that is the case is not entirely understood. There are lots of interesting models for it, but I'm not gonna get into the weeds on that one. And so so rapamycin, and again, the molecular biology associated with it is that it very clearly has effects that are beneficial to enhancing health span and lifespan. Although, in general, what's been found for human populations is that there are some negative side effects to it. So as a lifelong course of action, this is not something that people believe you should be taking right now as a way to extend your life, because then there can be health consequences where you actually don't get the benefit. Yeah, Janelle makes a very important point, which is we are not mice. And so that's a key part. So I want to mention at this point too, there's another compound known as metformin, which was a drug that was, again, I don't know its origin, but was used to treat diabetes. And again, the same thing, when people are looking at mouse lifespan extensions, they discovered that it does work for that. And so, again, through a lot of molecular characterization, they figured out what the targets of metformin are. And then also you'll see in the diagram that's here that mTOR is one of those downstream targets for what metformin is doing within the cells. And so, again, this is one of these interesting points too, is that, if you have a compound that's effective at doing something about diabetes, and metabolism and sugar metabolism are relevant to aging, then it kind of makes sense that the actual specific mechanism for how it's treating diabetes is that's also doing stuff that helps with anti-aging. Although, again, those types of things usually need to be sorted out more specifically. So metabolism is a driver. It's related to the aging process, although, again, probably not through the reactive oxygen species. And interventions that affect metabolism have an effect of improving lifespan. So another major topic, another major driver of aging is this concept known as senescence. And so senescence as a cellular concept has been around since the 1960s. There was a researcher, Dr. Hayflick, and what he defined was something known as the Hayflick limit, that if you just take normal cells, put them in a culture, and let them grow, they eventually just stop growing. After about 60 or 70, about 126 divisions, the cells just stop. Now, they don't die, they don't blow up, they just sit there, not do anything. And that became known as the Hayflick limit. And what I'm showing here on the left-hand side is a more recent paper re-examining this, but demonstrating one very important concept, is that if you add back an enzyme that allows telomeres to be replenished during cell divisions, then this Hayflick limit doesn't happen. And so the key point here is that telomeres are ultimately something that induces senescence as they get too short. Now, the program of senescence is something related to short telomeres. It's a cellular program to say stop growing. There's things that say, hey, something's going on here, so we need to senesce. And what I have here in the right diagram is from a review describing how other things can impact cells to drive and create senescence. And that includes damage to the DNA. That includes mutations that are oncogenic. It also includes reactive metabolites. So again, all the stuff that's happening, if it's at too high level, will cause cells to slow down. So those reactive oxygen species can create senescence. High mitogen signals, as well as proteotoxic stress. So if something's happening with the proteins that's causing them to misfold or not be replenished by lysosomes, by autophagy, then cellular senescence is something that can be driven by that. And so something that was discovered later, much more recently in the, I think about 2008 or so, is that not only does the cell senesce and decides it's taken itself out of the growth and the metabolism pathway, it also has something known as SASP, which is a senescence-associated secretory phenotype. And what's happening here is that neighboring cells can also basically go into senescence because the nearby senescent cell is saying, hey, you need to stop growing too. Now, all the details behind that are not clear, but you can imagine that if you have a body that's now developing a bunch of senescent cells and it's telling other cells to be senescent, that is something that can drive an aging process and the breakdown of cellular processes and for the body. And so here's the full picture from this diagram. And that the idea here is that when you think about senescence, that when you're a young, healthy body with metabolism that's saying grow and get to reproductive age, and you haven't had enough time to accumulate damage in cells, those exist, they're present, they're at a low level, it's not a big deal. But then once you pass the threshold of how many senescent cells you have, that can start driving chronic diseases or dysregulation. And so the idea is that if you can clear out and remove senescent cells, then actually you can get rid of all those disease states because you're helping clearing out the cells that are causing them in the first place. And so in the very far diagram, this idea of a senolytic intervention, again, a drug or some other treatment that takes senescent cells and tells them to lyse, in other words, to blow up, then that can come back. All right, now, does this work? So if that theory of if you can clear out senescent cells, does it actually help de-age something? And so here's an example of something that's well, it looks like a very complicated set of data, and it's a lot of work that goes into it. But there's actually some very basic trends to look at from the graph on the bottom side. And what the researchers did here is they recognized that there's an enzyme called lysosomal beta-galactosidase that is specifically activated in senescent cells. So they created a compound that enzyme acts on it, and if that enzyme acts on it, it creates something that kills the cells. And so what they're showing here basically in each one of these graphs is an example of how well the mice are performing a physical task. And on the very left-hand side, you have young mice, then middle-aged mice, and then old mice. And you can see that for most of the graphs, the average, the cluster, is going down. But if you take old mice and create a compound that's clearing out the senescent cells, that's what you have there in pink, you'll notice that those averages go back up. And so this is an example of how this type of intervention, as you measure various objective ways of understanding, say, frailty or some other index of an aging process, then this intervention helps alleviate that aging process. And so the idea that senolytics work then is something that also does make sense for how we understand some of the drivers of aging as well as interventions that can help with that. And so people are trying to find what are so-called senolytics, where you don't have to have that are like this that end up having broad applicability that target very specific cells in the body. Another driver of aging is inflammation. So inflammation is the type of body response that you have to infection. So when you think of fevers or cells trying to mobilize to repel a virus or a bacteria. And just like, and then there are also very specific subcellular processes that are inflammation responses as well. And just like if you have a cold, you don't want to be walking around with a fever when you're not being attacked by an invader. Having inflammation is great to have on a short-term basis, but it's not good in a long-term basis. And what this graph is showing is that one of the key regulators of this is a gene known as NF-kappa B. NF-kappa B is something that tells other genes to turn on. And that there's a whole complex set of things that's happening from an inflammation response. And that inflammation response is that because you're setting up to try and repel an invader, all the stuff this is like a nation going to war, that all the normal domestic economic things that you would have during a war don't work as well. And so combating inflammation is something that's been recognized for a long time as something to try and tamp down, even in the aging process. And so what I'm showing here on the left-hand side in terms of interventions that can help with inflammation are that there are other genes in the cell that tell NF-kappa B to be on or off. And what you see here on the left-hand side is another one of these lifespan measurements of mice, where if you are a normal, untreated, non-intervention mouse, it's that red line, and that's your death curve in this particular case. If you're activating NF-kappa B by adding IKK beta, then you actually decrease the lifespan. That's the blue graph. And then the green is showing that if you're inhibiting NF-kappa B, then you can get a lifespan extension because you're now inhibiting the inflammatory process within those organisms. Now, again, these are caged mice. It's good to have an inflammation response. You wouldn't necessarily want to walk around having that always turned off because then that makes you susceptible to diseases. So, this has to be something that's very targeted and clear. But this group also showed something very interesting, is that when they were looking specifically at overactivation of the immune response in the hypothalamus, which is important for making hormones and regulating hormones in the body, that those decreased. And if you intervene by adding gonadotropin-releasing hormone, basically injections back into the mice, that those helped alleviate some of these aging effects that you see when they are being inflamed. And so the idea here is that there are some very important ways in which inflammation can degrade cellular processes, the hormone signaling, what the body's doing. And in fact, an intervention can be to make sure that you are compensating for those processes. Like in this case, adding back more hormones that are being depleted. So Shiloh brings up an important question, which is, I think, when you go to the doctor and you have your blood levels measured and other stuff, there's lots of things that are markers of health, and sometimes those markers of health are also related to the aging process. But again, they aren't necessarily diagnostic for how old you are, what the aging is, whereas something like methylation is maybe potentially a little more insight into specific aging processes. But being healthy is all a part of this, is that you want to make sure. There are times where if you have some part of your system that is compromised, that can actually spill over and drive other further sickness or health problems. Like having diabetes is something that can lead to cardiovascular problems. Okay, so another key driver of aging that's been appreciated for a long time is damage to the DNA. And this has been, the insight from this came from progeroid syndromes, and these are syndromes in which a young person ages dramatically. So you see on the top left-hand side, there's a Werner syndrome patient at the age of 15, and then at the age of 48, she looks a lot more like a pretty frail 85, 90-year-old. Another Werner syndrome patient on the right, same thing, is that at 13, they look they look normal, but by the age of 56, they look like they're 90. And what's also associated with these progeroid syndromes are, they typically die early because of some sort of tissue failure or organ failure, like heart attack. There are a wide variety of these, and what's important is that the genes that underlie these syndromes are things that are known to be that are important for repairing DNA. And so one particular type of DNA damage is known as the double-strand break. So DNA has two strands. If you break through both of those at the same time, that's a double-strand break. And so the failure to repair these is something that, at least in these progeroid syndromes, is clearly associated with creating aging phenotypes. Although how well that's related to the general aging process has not been clearly delineated up until, I'd say, the relatively past 15, maybe 10 years. And here's an example that the research in this case is showing that if you look at post-mortem samples of people diagnosed with Alzheimer's disease or people that were diagnosed with mild cognitive impairment, MCI, but then post-mortem, that they have some degree of Alzheimer's degree onset. If you actually look at those cells from those post-mortem tissues, those little green dots represent double-strand breaks. And that's because they're using an immunofluorescent staining to a protein, then those proteins associated with double-strand breaks. And this graph is showing that compared to control tissue samples, there are lots more of these double-strand breaks that are present in the neurons or the astroglia of these MCI or Alzheimer's disease diagnosed individuals. And so there's this association double-strand breaks with leading to aging type phenotypes. And another set of research on the right that I'm showing here is that when you look at female human tissues, they start showing a decrease in the ability to repair double-strand breaks. And so the chart graph is showing how quickly things are getting repaired, and the fact that young donors, a lot more is repaired quickly and early compared to age donors. That's an example of that. Okay, so the last kind of major theory of aging, in terms of how we understand the process at a cellular level, is something known as the loss of epigenetic information, or also known as the information theory of aging. And this comes back to the idea where I talked about before with methylation, is that certain genes in the cell are supposed to be on, certain genes are supposed to be off. And if that gets dysregulated, then the cell may not know what it's supposed to be doing. And so this graphic is showing here, this is A, B, and C are nicely organized. The chromatin, the DNA is nicely organized by the proteins that package it up. And on the right-hand side, you're showing that I'm showing that that's not the case. And so this theory makes a lot of sense. And in fact, one reason to think that this is a really important way to think about cellular aging is related to the fact that a diagnostic assay for disorganization of genes, methylation, ends up being one of the best predictors for cellular aging. And what I have here, again, this is another big, large graph, I know, but the key point is that if you reset the cell's differentiation, and this is the Nobel Prize, I think in 2012, was for Yamanaka, and what he discovered was the ability to basically take adult cells and turn them back into stem cells, so induce pluripotent cells. And those Yamanaka factors basically de-differentiated an adult cell, a skin cell, back to being like an embryonic cell. And what these researchers are showing here is that if you basically do something for a very short amount of time, where you just say, let's just gently prod these to be a little bit younger, then you can alleviate and help reset the methylation patterns. And so these graphs are showing here different types of cases for different genes and different ways of looking at membrane potentials, mitochondrial, reactive oxygen species, the health of cells. Is on the left-hand side, you have young cells that are blue, you have old cells that are red, and so you notice red is usually on average lower than blue, but that aqua color, this is the cells that have an intervention to try and help reset their methylation to a younger state. You notice that that's coming back up more towards the blue, and so that's an example of this intervention that works. Another thing that's important to recognize, and this is work by David Sinclair, who's one of the key people driving a lot of this information theory work forward, is that if you just induce double-strand breaks using an intervention, they're using an enzyme to make double-strand breaks, that you see the same loss of information within the cells. And so the model here is known as ICE, so basically the induction of chromosomal damage. And what the right-hand graph is showing is that you take normal cells, their epigenetic age is about what you expect them to be. But once you start inducing double-strand breaks, then their epigenetic age skyrockets to be really high. And so one thing that's important, one thing that this experiment was trying to differentiate, is that it's the creation of double-strand breaks, but not necessarily the creation of mutations. Because one of the reasons people thought people aged was the accumulation of mutations. And in fact, it's actually double-strand breaks that may help and be a matter of resetting the epigenetics. And this makes sense. The reason why this model makes sense is that if you're making a break in something, well, if you have that within a package, if you have that within a packaging, you need to remove the packaging to repair it. And then the efficiency of putting that packaging back is problematic. It's like if you're looking, like if you want to know what somebody's sending somebody else in an envelope, well, you can open the envelope and see what's inside, but to make it look like you never opened that envelope, that's not as easy. And so that may be what's happening on a cellular level. So in terms of kind of putting this together, what a lot of the research has been showing over the past 15, 20 years from this molecular and cellular biology level is that genomic instability, inflammation, and this loss of epigenetic programming can be very important drivers that may even be the initial initiating causes. But in each case, when you look at any one of these factors that you're driving, it tends to drive the other factors forward as well. And so there's a good and bad part of this, is that one, the bad part is once you start driving one of these processes forward, it starts a snowball effect where there's feedback that causes more of that problem. On the other hand, and I'll talk about this a little bit later, it also means that if you can target one of these, in some cases, it actually helps reset the rest as well. So what are the consequences for this? How do we want to think about that? I've seen some questions like, well, how is this important for the clinic, or what can I do? Well, what we're seeing right now is a large number of people developing research and proposing clinical trials where within various types of aging, chronic diseases, or even trying to prevent certain chronic diseases, can we intervene with removing senescent cells? Can we have compounds or gene therapy that reduces inflammation? How can we reduce genetic instability? How can we reverse the programming of the cells? The idea of putting in transient Yamanaka factors to rejuvenate cells and tissues is very much at the forefront of the research that's going on here. And so, but then maybe that's not the key question, right? Because I mean, so certainly if you have something, you want to keep an eye out in literature for whether there are interventions where if you're at the hospital or you go to the doctor or your annual physical exam or the things that they can say or recommend. Right now, you can't go request these. These are not something that as a patient you can go to the doctor and say, hey, give me senolytics. So in terms of what can we do? Well, one of the interesting key parts of this, and this is one example, there are others, but that this compound NAD seems to be a key metabolite in helping have healthy inflammation, healthy DNA repair, healthy epigenetics. And so the concept here and what's known is that your levels of NAD go down as you get older. So what's driving those to go down? It's not exactly entirely clear, but once those levels start going down, it's like I said before, that can start driving further aging forward. And so what I have here are some examples of some research where they show that if you basically give mice a compound that helps increase their levels of NAD in their body, then you can see an actual increase in the NAD. So those are the red bars compared to the black bars. But if you look at the lower graph, this is one example of a phenotypic effect where if you're measuring how well they can run a treadmill, then if you take age-matched mice, the ones that have higher NAD levels are running better. And so again, the idea that here is a metabolite, something that can be influenced by diet or maybe supplements, is an idea of something that is a intervention that we can participate in. And there are compounds out there where people say, hey, there are things that a little bit act like senolytics. There are these things that are slightly anti-inflammatory. Again, any tea that comes out these days, they're trying to tell you how it has this health benefit for this or that. And so what I want to describe here is that when we think about interventions, there's what's known as the geroscience hypothesis. And that when you think about your diet, when you think about these compounds, or things that might be analogs of them that you can have in your diet, then these are things that may be inhibiting fundamental aging mechanisms. So I've talked about some of these, the DNA damage, epigenetics, reactive oxygen species, what's happening in metabolism, that there are compounds that have these effects just from normal everyday eating or might be available as a supplement. And so if you can inhibit those fundamental aging mechanisms, then you can help stave off the aging phenotypes, and you can have benefits of a healthier, longer life. Can they get to the point necessarily of rejuvenating you? Probably not. Again, the ability to rejuvenate probably mostly comes from things that are molecular biology interventions. But this last diagram here is just trying to integrate this into the whole that, when you think about exercise, having low fat tissue, adipose tissue, a healthy diet, and calorie restriction. Again, calorie restriction in particular, these are all things that have been shown to boost the body's NAD levels. These are things that have been shown to reduce inflammation. And when people measure methylation in people doing exercise or different types of diet, then these are things that help with the methylation. And that's actually, that's one thing to be very fair about, is that if these types of compounds can help from a physiological measurement point, make your methylation patterns younger, and this is something Syzygy is mentioning about NMN, NMN in the chat, is that this might actually be an intervention that does, in a sense, rejuvenate. Now, the thing that's limiting is that you aren't necessarily getting that compound into every tissue. But again, if you make some tissues healthier, then adjacent tissues can become healthier. So there's a lot that can go on. And that's one of the key points, is that when you think about these interventions that help with basic cellular processes, once you start helping one process, the reason why you can look at a measurement of NAD and say, oh, we're also associating this with better inflammation, better autophagy, better nervous system, a decline in aging phenotypes, is because these are all integrated as a whole. Okay, so in summary, the last 20 years have been pretty amazing for understanding the basics and physiology of the aging process. We have a lot more about the ideas of what aging is at the cellular level and how that relates to tissue in the whole body. And what's wonderful is that they're not mutually exclusive. These are all things that seem to be related. In animal models and human cells, and to some degree in clinical trials, there are compounds and interventions that can slow the process of aging and can also reverse aging. Now, again, how much of these are true things that can be effective and worth your time and money to do as an individual is, I think, still under investigation and not entirely clear cut. But that what we do have now is that we think about our lifestyles, we think about what we do in terms of healthy eating, exercise, what types of food we eat, what types of activities we engage in, is that this new cellular understanding helps reinforce and provides the rationale for the concept that healthy living is the best way to stave off the Grim Reaper. So with that, I thank you for your attention. I'm here and happy to take any sorts of questions, and hope you enjoyed. So there's a really interesting question in the chat. So I'm happy to take some questions, and I didn't quite time my talk right, so I'll stay around a little bit extra. This question that Roy asks and Sizu G brought up, should I take an NAD supplement? And I will say from my own scientific point of view, I'm not sure that there's been enough high quality, gold standard, double blind studies of large scales of people to say that spending, say, $2 a day gives you, after you spent several $1,000s buying them, $1,000 in benefit for your lifespan. What I will say is that when you look at NAD supplementation in various clinical interventions, and there are clinical trials that are ongoing, and so you might want to sign up for a clinical trial. You can always do that. There does seem to be some value in de-aging benefit as you measure it. The one thing I will say about NAD supplements, as you look at most of them, they all have very high safety profiles. They are not shown, compared to the benefits you see in human trials that are short term, the downsides of them are incredibly minimal. And so, the opportunity cost there is, are you wasting your money on something that doesn't help you very much, and you could use that money for better purposes? However, I think, if you overall look at what happens with clinical trials and the data you have, again, these are clinical trials on healthy people. They're signing up healthy people to see if these have these de-aging or stave off aging effects. You could. Well, so wisdom brings up an important point, and that's why it's really important to do really good clinical trials that are also being managed and run by independent agencies with a lot of transparency in the data, is that placebo effects can affect and mess up results. Yeah, I think people have shown that you can have 5 grams of NMN per day without any sort of negative health consequence. But then how much benefit, that's going to cost you a lot of money.
Stephen, I wonder if you saw my question.
Which one was it?
If at an advanced age one has had no major diseases and has a normal blood panel, what does that indicate about these biological considerations?
It's interesting. That's actually a really interesting question, because one thing I didn't delve into very much today is that there's been a lot of research that has looked at the genes of long-lived people. And there are genes and alleles of genes that are associated with people who just naturally live very long lives, these centenarians. And so in many ways, what's interesting is, taking supplementation may not help people who are already pretty healthy and have lived long, good lives and have, eat healthy. They may have, the amount of effect they have might not be very dramatic compared to someone whose genes are kind of driving them to an earlier aging phenotype. On the other hand, one thing that's very clear, this accumulation of damage model means that every little bit that you can push the metabolism towards less aging, the earlier you do it and the longer you do it, then the more benefit you can get in the long run. And so, I think at whatever age you are, and whether you're healthy or not, there are very good rationales for doing healthy things in terms of your diet, your exercise, and potentially with supplementation. Does that make sense? Does that answer your question?
No. I'm asking what you can, are there any assumptions you can make in looking at a totally normal whole blood panel, blood labs, and the fact that one has had no major diseases in and correlating that with the stuff that you're talking about?
Yeah. Well, so I did kind of answer your question, which is that one of the inferences we have now in terms of long-lived people and the genetics that help them is that health span is very much related to lifespan. So if you are a person who has really just traditionally not gotten very sick, not had any major diseases, that's probably due to some genetics that has helped you get there in the first place.
Mm-hmm.
And so we are learning about this, and that is lots of research that's going on, is trying to take populations of people, correlate their methylation clocks with their chronological age, with their lifestyle and their how healthy they've been, and trying to understand those genes that are involved with that. Can you infer anything from that? Again, from an individual point of view, that's hard. You might have just been lucky. That's the other aspect of it too, from an individual.
Okay.
Yeah, I mean, but I still want to just reiterate too, is that, if you've had a healthy, you might think you're lucky, and you might be very lucky, but no matter what's happening, that you've had a healthy life, this accumulation of damage and hitting these thresholds, these are things that are inevitable for everyone under normal living lifestyle conditions. And so there's always this inevitability of the aging process, unless you're doing things that push it to rejuvenate or almost be at a standstill. And those are almost always very specific interventions you have to do with how we understand aging these days. So I see a question. Does that answer your question?
Yeah, well, I was surprised when you said that antioxidants are kind of moot. I was interested in, because I read research about astaxanthin, which is a very powerful antioxidant. And from what you said, that doesn't matter that much.
Well, okay, so let me also just make a quick clarification about that too, which is, as a driver of the aging process, reactive oxygen species does not seem to be at the level they're present in normal people to be a clear driver of that. Now, the thing is, an antioxidant, the actual compound you're talking about, just like I talked about with metformin and rapamycin, is that compound, if it's known to be helpful for reactive oxygen species, it actually could have other effects on the body too. And in cases where there can be, like in mitochondrial dysfunction, you can have an uptick in reactive oxygen species, and then those reactive oxygen species can drive inflammation. Inflammation is very sensitive to reactive oxygen species. And so one thing to keep in mind is that there may still be spaces and interventions in here where you're targeting and you have compounds that are known to target reactive oxygen species. And in a normal physiological healthy state, that doesn't do much. But in certain cases, it may actually be helping with an overabundance from another mechanism that's creating problems. And it could help intervene in one of these main drivers. And then the other possibility is that the compound just actually has a target that people don't appreciate. So, if people have done clinical trials where you've had, double-blind studies, people have done efficacy profiles of any given compound, you can still have an effect. They can still be beneficial. They just may not be for the reason that they're being advertised. If that makes sense.
Okay,
So,
thanks.
yeah. And so there's an interesting question from Shiloh here, which is, Japan, in general, as a nation, and the people with that Japanese heritage tend to have longer and seem to be healthier lifespans and health spans than other people. Then why that's the case, we don't know, Shiloh. Now one thing that's interesting, and I'll talk about one supplement from a researcher who's done a lot of mouse model stuff, is a compound known as fucodin, which is actually derived from seaweed. And so that's something that's been shown to have anti-aging properties. And so again, one thing that could be happening is that because there's a healthy thing and something that people eat at a high prevalence, then there's this environmental impact that's helping tamp down on a major driver, fundamental aspect of aging. But the other aspect of it could be too that they do have, again, we recognize them as a slightly different heritage because they have a slightly different gene set that leads to an obvious phenotypic difference. Well, there could just be other genes being carried along where they just in general have better longevity genes, or maybe in general they have better anti-inflammation genes. And so we don't know that. And those are things that take a lot of time and effort and money to parse out. So, why they're different, we don't necessarily know. Now, I will mention one other thing too, which is this idea of what so-called blue zones. And I'll just type that really quick. If you do a Google search for blue zones, there are subpopulations of people like Mediterranean. There's a community out in California where as a whole, these are people who live healthy lifestyles. They exercise a bit, they eat healthy, they have very, very low amounts of processed foods. Again, there are certain processed foods that we know drive a lot of problems with the body, especially with inflammation. That you have pockets of people that, irregardless of their heritage and genetics, clearly live healthier, longer lives. And so there's a lot of interest in trying to understand that. Scientists are trying to break it down, doing their reductive process and trying to understand that better. But I really can't comment too much on it other than there are clear examples where living healthy lifestyles helps with health span and lifespan. And one thing that people are trying to do now is by not only just recognizing that from a phenotype, but now going and saying, what's their methylation clock? What are their inflammation markers? Trying to study these people in a rational way to get even more insight to what's happening at the very basic level. Oh, Thunder mentions that there are cookbooks that these blue zone people are putting out, or people who are studying them are putting out cookbooks, so something to maybe keep an eye out for. All right, so we've hit 10 minutes after. I will stick around for another 5 minutes, but otherwise, I want to thank you for sticking around, for your attention. I want to thank Wisdom again for the opportunity and to have this conversation and talk and update, and I hope you learned something. There will be follow-ups. I already have in mind a follow-up talk at some points in the future, and so thanks for coming.
Yes, I want to thank you so much. Stephen was the reason this whole thing that is kind of ballooned started. He offered to speak to the Whole Brain Health, and I thought, gee, we want to get an audience for him, and so we started doing that, and before you know it, we've got over 50 events here this week. So please be sure you get the list, the schedule, And Stephen, I look forward to the next installment.
Thank you, Wisdom. Have a good day, everyone.